Please welcome back Melinda Bachini.
I love that voice. Welcome back, everyone. I hope you all had a good break. We're going to get ready for our next panel, and I'm going to call up the next participants for the next panel, Dr. Ryan Fields, Caitlin VanLith, and Rebecca Auer. If you guys could come to the panel, or come up to the stage, to the table, and we will get started. Dr. Ryan Fields is a surgical oncologist and translational scientist who treats solid tumors, including pancreatic cancer, primary metastatic liver cancers, gastric cancer, soft tissue sarcoma, and melanoma. His research training began at the University of Michigan under the mentorship of Dr. James Mueller and Alfred Chang in tumor immunology. These early studies of dendritic cell-based cancer vaccines set the stage for his interest in cancer biology and immunology. And since that time, he's continued his research and clinical training in surgical oncology and solid tumor biology and immunology. He is the chief of surgical oncology and serves as the director of Washington University Solid Tumor Tissue Bank and Registry. He also is a co-leader at Barnes Jewish Hospital and an Alvin J. Siteman NCI designated comprehensive cancer center at Washington University School of Medicine in St. Louis, Missouri. Thanks, Dr. Fields.
Thank you, Melinda and Stacy and everyone for giving all of us, Caitlin and Rebecca, a platform to really talk about, I think, one of the most important things, and that's some of the research that we're all doing that directly involves patients as part of the research. So it's a pleasure to be here. I'm going to give a bit of an overview about our Cancer Moonshot program. Cholangiocarcinoma, it's got a bad NIH name, the Participant Engagement in Cancer Genome Sequencing, or we call it PECGS program. The study goals really for this program is to focus on cancer disparities and rare cancers with rigorous patient engagement and partnership with world-leading genomic sequencing to fill knowledge gaps. And that's the NIH's definition of really trying to involve patients in this research or transforming patients into partners. Oh, I think you got, this is the wrong slide deck though. This is, I don't know if they can put that, this is for our 5 o'clock session. Sorry, we had 2 talks. While they're pulling up the other slides, by way of background, the Cancer Moonshot program, many may remember, started many years ago when then President Barack Obama announced the initiative. It essentially infused an additional $1 billion into cancer research. So of course, when that happened by executive order, first order of business was really figuring out what to do with that money. And so they assembled a blue ribbon panel, and that blue ribbon panel came up with a number of initiatives. One of the initiatives was to more directly involve patients in research, and in particular, research that had a genomic focus to it. And the reason for that was, if we looked at many large-scale cancer studies, they weren't very representative of the patient population in terms of demographics, meaning they were overrepresented typically by Caucasian populations, more urban than rural populations, a higher socioeconomic status, things like that. And there was a lack of study and participation in a lot of more rare or underrepresented cancers like cholangiocarcinoma. So one of the funding opportunities out of this Cancer Moonshot was this program that I'm talking about, our Participant Engagement and Cancer Genome Sequencing project. So part of the goal of this is to enroll patients with cholangiocarcinoma. We've partnered with Melinda and Stacy. Melinda's on our external advisory board and has been integral to partnering with the foundation. And so what it involves is talking to patients with cholangiocarcinoma and enrolling them into the study. They can be anywhere in the country, and the patients, we then, with our team, incorporate all of their medical records and data. We obtain their previous tissue biopsies and obtain their previous surgically resected specimen, anything that's available, to study and deeply profile these cancers using cutting-edge, state-of-the-art omics, some of the things you've heard in some of the other sessions. But a large part of the study also involves our colleagues in public health sciences Two of the co-PIs on our study, Bettina Drake and Graham Kolditz, are world-renowned social scientists, public health scientists. And the goal of that is to better understand why patients consent to clinical trials, why they maybe won't consent to clinical trials, and what their perception of value in participating in a clinical trial is, and how we can enhance that value in terms of returning results, discussing with patients the results, and the patients and their caregivers being more directly involved in that research. So to date, we have enrolled over 200 patients with cholangiocarcinoma, many who may be in the audience today and are participating in our workshop a little bit later today, where we're going to be continuing to do some of our focus groups and survey work to, again, try to address these important questions. A couple of the slides that I were going to show is, we're about 2.5 years into this project, into a 5-year grant, so we're starting to get enough patient samples into the pipeline to be able to study and profile some of these tumors. One of the techniques that we've used that I was going to show a couple of slides on, it involves a platform called spatial transcriptomics. It's one of kind of the newer cutting-edge ways to really deeply look at a tumor in a genomic sense, but also preserve some of the architecture of that tumor, so we can literally see what cell's sitting next to another cell, a cancer cell, say, next to an immune cell, and what that immune cell is thinking or doing. And we've seen a couple patterns in patients that have cholangiocarcinoma that don't develop disease recurrence, and those that do with some of the therapies that they're on, in particular some of these immune-based therapies. And so we think that there may be a signal there that we can exploit, or we can better understand mechanisms of response and resistance to some of these immune therapies and targeted therapies. So it's going to be very complementary to some of the other work that you've heard and some of the other talks that you've heard from in some of the other sessions about taking a signal that we see, meaning response to immune therapy that's in a small cohort of patients, and hopefully expanding that. When you walk out the door to the right, we have a table where Danielle Grasso, a nurse that's leading our project, is sitting along with some of the other members of the team. So for whether you're a provider, a caregiver, a patient, if you would like more information about the study, potentially enroll in patients in the study or enroll yourself, we've got a lot of information sitting there. I'm here through the rest of the conference as well. You'll see me walking around. I'm happy to talk with anyone at any time about the project, and happy to take any questions once we have the panel as well. Thank you.
Sorry, Ryan.
That's okay.
Sorry about the slides, but we'll do a webinar when we get home and update everybody. Okay, next up we have Caitlin VanLith. She's going to talk to us about the genetic susceptibility to cholangiocarcinoma and give us an update on GWAS. Caitlin is a fifth-year MD PhD student at Mayo Clinic. Her thesis work is on the epidemiology and discovery of rare genetic variants in cholangiocarcinoma patients. She is part of the team from Mayo, coordinating a global genome-wide association study, the GWAS, of cholangiocarcinoma with the ultimate goal to better identify patients at risk for cancer development. And through her work, she has been able to work closely with the Cholangiocarcinoma Foundation, the West Africa Institute for Liver and Digestive Diseases, and the Africa Hepatopancreatibiliary Cancer Consortium. Caitlin hopes to build her career using collaborative networks to identify risk factors for disease development and to increase representation in scientific research. Thanks, Caitlin.
Thank you, Melinda, for the introduction. So as Melinda was saying, my name is Caitlin VanLith. I'm a student at the Mayo Clinic in the lab of Dr. Lewis Roberts, who just caught a flight out of here. But we have a big team from Mayo here that will be happy to talk to you about anything that I'm going through today. So you may have heard GWAS going around. You may have seen the sign in the hallway. This is a project that we're doing collaboratively with the National Cancer Institute and the Cholangiocarcinoma Foundation in order to discover variants, genetic variants that are associated with cholangiocarcinoma development. So today in this quick presentation, we're going to go over cholangiocarcinoma. I know that some of you are experts in this, and some of you are new to this area of medicine. So we're just going to do a quick overview. We're going to talk about what's known about the genetics of cholangiocarcinoma, and then I'm going to talk about the International Consortium for the Genetics of Biliary Tract Cancer, the work we've done and the work that we're still doing. So cholangiocarcinoma, it's a cancer of the bile ducts. There are a lot of different locations within the liver that it can present, intrahepatically or extrahepatically. There are some well-recognized risk factors. People have talked about primary sclerosing cholangitis, which is a rare autoimmune disease. And there are some other risk factors like colorectal cysts, biliary tract stones, and liver fluke infections, which is common in Southeast Asia. But most cases have no known cause. There are these risk factors, but most people don't have any of these risk factors when they're diagnosed. And that makes early diagnosis difficult, and that means that prevention and screening for cholangiocarcinoma are really key. This is a figure from a paper about the epidemiology of cholangiocarcinoma. The small numbers don't matter. Mostly what I want you to look at in this graph, in this figure, are which countries are represented. So you can see that we have data from the US, Canada, a lot of Western Europe, China and Australia, but all of those great countries are not represented in this study. So now we're going to talk about what we do know about the genetics of cholangiocarcinoma. When we're doing genetics research, there are really a couple of goals that we're trying to achieve. So one of them is understanding risk, being able to proactively sequence someone's genome, understand what their risk for developing a disease might be, so that we can diagnose early. Another goal is to identify biomarkers. You have been hearing all about those. Those can both be diagnosis biomarkers, they can be biomarkers within the tumor to help with treatment. So that's another goal of genetics research, as is developing targeted therapies. You've heard about that, some of that in the clinical trials session about using certain biomarkers, genetic biomarkers, to develop targeted therapies. So in this research, we're particularly looking for understanding risk and identifying diagnostic biomarkers. Cholangiocarcinoma genetics are complicated. There haven't been a lot of obvious genetic links. There are some cancers that you think about really running in families, and cholangiocarcinoma isn't typically one of those. There's low heritability within family structures, and a lot of the genetic risks that have been attributed are attributed to the risk factors as opposed to the cancer themselves. A lot of variants that have been found in cholangiocarcinoma are common cancer-causing genes. This is the hereditary cancer syndromes like Lynch syndrome, BRCA mutations, and those can be both germline and somatic. So germline genes are the ones that you are born with. Those are the ones you inherited from your parents. And somatic variants are the ones that you acquire throughout your lifetime. So your blood likely has your germline DNA, your tumor sample likely has, well, it does have somatic DNA. So we're really interested in this study on germline variants. And the genetic profile of what we do know about cholangiocarcinoma is different between intrahepatic, extrahepatic. There are a lot of molecular subtypes of cholangiocarcinoma, and so it's really important to understand not only the cancer as a whole, but each of these individual subtypes and how it presents in an individual person. But the major limitation that we have to cholangiocarcinoma research is that genetic studies have been in largely small numbers of patients. It is a rare cancer. Any single hospital or center isn't seeing a ton of cholangiocarcinoma patients in a year, which makes putting together these large cohorts needed for genetic studies really difficult. So that's what our international consortium is going to try to address. This is some previous data from a 2012 paper, just kind of showing that a lot of these genes, IDH1, KRAS, TP53, those are the ones that are associated with cancer generally, as opposed to being specific to cholangiocarcinoma. And this is a more recent paper that looks at pathogenic germline variants. So germline variants being from your blood, not your tumor. So things that might predispose you to developing cholangiocarcinoma, pathogenic meaning disease-causing. So in a cohort of 132 cholangiocarcinoma patients, 15.7% of them with intrahepatic CCA presented with this pathogenic germline variant, and 17% of extrahepatic CCA patients presented with a pathogenic germline variant. And so this genetic research that has been done has really kind of shaken up that idea of, is cholangiocarcinoma inherited? And And so we know, based on a lot of these studies, that there is a proportion of people who are carrying genetic susceptibility variants to cholangiocarcinoma, and we'd really like to understand that further. And this is also from the same paper. It's kind of just pointing to small numbers in genetic studies. So there were 132 cholangiocarcinoma patients total, but if you look at the count, the y-axis on this graph here, for each of these bars, we're looking at up to 6 patients total. So it's hard to do these large studies. It's hard to get statistical significance, which really allows us to extrapolate into the general population when we're only seeing 6 patients in each group. So now we're going to talk about the International Consortium for the Genetics of Biliary Tract Cancers, which we're organizing with a lot of you to be able to have these larger numbers of patients that are going to be needed for robust genetic studies. This is an effort led by Dr. Lewis Roberts at the Mayo Clinic. He's my mentor. And then also Dr. Catherine McGlynn at the National Cancer Institute, Dr. Chris Amos at Baylor, and Dr. Manal Hassan at MD Anderson. And so this is the first genome-wide association study of cholangiocarcinoma. I'm going to talk about what that is. So in this figure, you can see the red, oops, how do I go backwards? Oh, here we go. There we go. The red patients here are ones without cholangiocarcinoma. The green patients here are ones with cholangiocarcinoma, and the boxes are around a genetic variant. So the bottom patient in the red row or column has this genetic variant, whereas both the top and the bottom in the green column have this genetic variant. So 33% of non-CCA patients versus 66% of CCA patients. This is a very generalized example. So this would be an example of something that's different. So when we're looking at these genome-wide association studies, we're wanting to collect data on not just 3 people in each group, but tens of thousands of people in each group to look for those really rare genetic variants that might be predisposing someone to cancer. So over the past couple of years, we've completed a discovery phase of this study to kind of show that it is feasible to give us data in order to get grant money to be able to do this large study. So we did a discovery phase of 2,600 cholangiocarcinoma cases. And these are coming from 24 sites in North America and Europe. Like Dr. Fields said, and like the map that I showed you at the beginning of this presentation, North American and European samples are what are being insanely overrepresented in genetic studies. So in this discovery phase, it was limited to those groups because of sample availability, but that's something we are also trying to push the boundaries on in this study. In this study, we have 2 different genotypings that we're doing. One, we're genotyping over 1.8 million single nucleotide polymorphisms using the what is called the Global Diversity Array. So this is looking at a large group of variants that has been validated in not only those North American and European populations, but in a large global diverse population. And then we're also looking for novel protein coding regions predisposing individuals to cholangiocarcinoma through what's called whole exome sequencing. So you have a huge genome, but not very much of it, about 1% to 2% actually codes for proteins. So in AIM2, we're looking just at those protein coding regions and trying to discover variants in what some people, but not everyone, as we're learning more and more about genetics, would say are like the very high yield important areas of the genome. This is a distribution of the discovery phase data. Again, about 2,400 individuals from North American and European sites. So this is what's called a Manhattan plot. And so each of those dots is comparing a certain genetic variant in CCA patients versus non-CCA patients. The higher up it is on the graph, the more significant it is, the more likely it is to appear in CCA patients, but not in non-CCA patients. So anything above the red line has reached a point of statistical significance, so those are things that we're really interested in. Anything above the blue line is reaching levels of suspicion, so things that we'd want to investigate more. Generally, the larger the study size is, the more that true results are going to move up on this graph. So with a larger sample size, we're likely going to see more statistically significant genes. The arrow is pointing to a hotspot in an HLA region. So HLA is what's coding for an important part of your immune system that is very diverse in the human population because it's beneficial to people to have diverse immune systems if there is a pandemic. So this particular hit that we're seeing here is in one of those HLA regions. Dr. Young-Hun Han, who I think is in the audience, I can't see anyone because of the lights, but he is the one that did the analysis on this discovery phase and found an estimated heritability of 12%, which means that in this data, we can attribute about 12% of cholangiocarcinoma to genetics. So again, this is really validating that genetics are important in the disease and that this is something we want to explore further. He also found different variants for intrahepatic cholangiocarcinoma, extrahepatic, PSC-associated, and non-PSC associated. So again, it's important to look not only at the disease as a whole, but specifically at those subgroups as well. He found 5 novel variants, 1 overall in all CCA patients, 2 specifically in extrahepatic CCA, 1 in PSC-related CCA, and 1 in non-PSC-related CCA. This was not the slide deck that I put together for this patient session, but this is a little bit more details about that 1 overall novel variant that was found. The p-value you can see is really small. That's the statistical significance that's showing that there's a good chance this is really a true result. And that 1 overall hit is this RS number is how we label genetic variants. It's in, again, those HLA regions, part of your immune system, and it's been associated before in other cancers, such as cervical, kidney, prostate, and thyroid cancers. And a lot of these 5 variants were in those HLA regions. In now the expansion phase, we're recently awarded a grant by the US government, and we have a goal of sequencing 7,000 cholangiocarcinoma patients in the next 5 years. And we not only need cholangiocarcinoma patients, we also need controls. So we currently are working with 82 sites from 31 countries, including the Cholangiocarcinoma Foundation. I think a lot of you have already participated in this study, and we thank you a lot for that. And you all have the ability to. So this was going to allow us to look at even rarer variants. And we do have an emphasis on global genetic diversity and increasing representation in this study. So we're not only working with sites from North America and Europe, but really globally we're working with sites. And this is important because different variants are expressed in different levels in different global regions. So previous research has been overwhelmingly white. Most development has been happening in North America and Europe. But this global diversity array, as I said, is really created with a global population in mind. So these are the countries with sites that we are working with right now. So we're really still in the recruiting phase and working closely with all of these sites in order to try to get representation from them. And as I said, you all have the ability to participate in this study. At the end of the hall, there's a sign that says GWAS on it. It's Grand Ballroom J. We have a team of nurses and phlebotomists in there that are helping us consent patients and draw blood on site. And we are collecting samples between 7 a.m. and 3:30 p.m. For the next 2 days. So I really encourage you to participate. This is important work. It's a paying it forward type of study. And if you don't have cholangiocarcinoma, if you're a scientist, a researcher, a caregiver, a friend, we are also looking for controls. So everyone can participate in this study. And for people who are watching this virtually, or people in this room who aren't going to have time to do the blood draw at the conference, we are also doing consenting, and we will ship you a blood kit, and you can go to your doctor to get blood drawn for it, all costs covered by us. So this is the QR code for participation externally to the conference. I'd really like to acknowledge everyone in my lab and everyone who is a part of this consortium. And I'd really like to acknowledge and thank everyone who's here. You are the reason that we're doing the research that we're doing. This is really patient-driven research. A lot of the questions that I'm asking in my work are coming from questions you've asked, observations you've made. So please do keep speaking up. Come talk to us about your experience, any observations that you've made, because you are the most important people in this research. Thank you.
Okay, and last on the panel, we're going to have Dr. Rebecca Auer talk about the Canadian Cholangiocarcinoma Collaborative, or C3. Dr. Auer is the scientific director for the Cancer Therapeutics Program at the Ottawa Hospital Research Institute. She's a surgical oncologist at the Ottawa Hospital and a professor in the departments of surgery and immunology at the University of Ottawa. She holds a Tier 1 clinical research chair in perioperative cancer therapeutics and leads a translational research program focused on understanding the promotion of metastatic disease in the post-operative period. More importantly, she's also a caregiver to her husband who has cholangiocarcinoma and a great friend. Welcome, Rebecca.
Thank you so much, and thank you to the foundation, Melinda, Stacy, and everyone for the opportunity to speak today about this initiative. It was announced actually last year. We just got funding for the C3 initiative about a year ago, and so I'm really excited to show you our progress and what we've been able to achieve and what we're still hoping to do. So I start with this slide because it's part of the spirit of this organization, but as a cancer surgeon and a cancer researcher, I've always acknowledged that hope is really critical for patients, and research can fuel that hope. I have 3 boys. My middle son, when he was about 7, he came home from school and he said, "Mom, what's the most powerful word in the English language?" And I wasn't sure. I think I said love. He said, "No, it's yet, because yet makes everything that's impossible possible. If you add yet to the end of a sentence, you can do anything." And I thought about that all the time. And from that moment on, when my patients would ask me if they had what was considered an incurable cancer, they would say, "Well, Dr. Auer, is my cancer curable?" I never said, "No, it's not curable." I said, "It's not curable yet." And I truly believe that research is the power of that yet, and research really gives us hope. And I have to be honest, about 2 years ago, when my husband was diagnosed with stage 4 cholangiocarcinoma, I had what I call my steep left turn. And I kind of, even though I understood how important hope was, I really didn't feel that I had much. And this picture here is because I spent hours online looking at people like you. Many of you are here again today with your signs that said survivor, 2-year survivor, 5-year survivor, 10-year survivor. And I looked at those pictures until I believed that it would be possible that my husband would be one of those people. And really the hope that I was able to find was so in such large part because of the foundation. John Bell is a very well-known scientist. He also happens to be my mentor and works at the Ottawa Hospital Research Institute. And this is a quote that he said a number of years ago. But a couple of days after Adam was diagnosed, I went up to my lab to gather some stuff. I was going to take a bit of time away. And I stopped in his office to tell him what was going on. And I remember he kind of looked at me and said, "You know, all the time you tell me that, all these amazing things that happen, these patients that you looked after, and they're on these drugs, they didn't even exist when you did your training in New York, and and now they're cured. And, we all know it's just a matter of time. And it's just a matter of which cancer will succumb first. And I guess we have to start working on cholangiocarcinoma now. And I said, why don't you, go and take a few days off, but we're going to have to get to work. And he actually said to me, remember Melinda? And it was crazy because maybe 5 years before Adam had been diagnosed, I had received a travel fellowship to go down to the NIH. We were thinking of starting a TIL program, the tumor-infiltrating lymphocyte that Melinda had received. And at that time, the only thing I heard about was Melinda, because it was right around the time that her famous story came out in Science, and I probably spent 30 hours in rooms talking about Melinda's T cells that I'd never even met her. And of course, I emailed her that night, found her online and emailed her, and I'm sure anyone who's reached out to Melinda from the patient or caregiver side this story will resonate. Like within a few minutes, she emailed me back, and she said, you have to have hope. And she was right. Just a few things that, these are some slides that I used to give in talks as the director of cancer research to try and explain to people why it's so important to invest in cancer research. But truly, there has been an unbelievable explosion in technology, and that explosion in technology has allowed us to look under the hood of cancer cells and really understand the molecular underpinnings of how those cancer cells interact with our body, with our immune system, with the rest of our normal cells. And just a few facts. Think about this, that it was 2003 that we sequenced the first full human genome. That's only 20 years ago. It took 13 years to do it, and it cost $2.7 billion. 20 years later, we can sequence a genome for about $1,000, and it takes between 1 and 2 days. That's a huge transformation in what we can do. CRISPR-Cas9, you've probably heard of as gene editing technology. It was only discovered 10 years ago in bacteria as like a complete basic science discovery. And just last year, a therapy was FDA approved using that technology. So the pace of discovery is unreal. And the same is true with immunotherapy. It's only been a decade that we've had immune checkpoint inhibitors, and they've completely transformed the way we think about cancer. And when we think of cellular immunotherapies like CAR Ts that you've heard about, and T-cell infiltrating lymphocytes or TIL therapy, really the first FDA approval was in 2010, which is, again, not that long ago. And now there are 26 approved, but 7 of these were approved in 2023. Again, the pace of discovery is unreal. So where are we now? Maybe with cholangiocarcinoma, we have 1 or 2 dozen targeted agents and directed immunotherapies, but the future is a huge, vast number of these things, and our challenge will be in figuring out when and who to use each one for. And you can see this reflected in the number of FDA drug approvals per year. And so I really did spend some time thinking about that, and I guess, as my sister always says, you fake it till you make it. Eventually, I did come to the place where I felt like there was a lot of hope, and I wanted to bring that hope to Canadian patients. So with John Bell's mentorship, we got together a group of people, and myself, and one of my colleagues, Dr. Simon Turcotte, who was actually one of the clinicians that looked after Melinda at the NIH, and is a Canadian leader in TIL therapy, and then Carolina Ilko, who's an amazing, brilliant scientist. And we put together this proposal, and we really wanted to do 3 things. We wanted to make sure Canadian patients had access to better care so that they too could have hope, and really inspired by the Cholangiocarcinoma Foundation in that aim. The second one was to be able to offer TIL therapy or T-cell tumor-infiltrating lymphocyte therapy to Canadian patients, which is not available in Canada at all. And then the last one was an immunotherapy pipeline for cholangiocarcinoma, so that we could be on the preclinical side and the pipeline discovery side. We were successful in obtaining a $7.4 million grant for this initiative through the Canadian Cancer Society and CIHR, which is sort of the equivalent of our NIH, and that was last year. And the one icon I don't have on here that really should be on here is the Cholangiocarcinoma Foundation, and especially the Canadian Patients advocacy and support group, because really they were at the table from day one. It was actually at one of our Canadian advocacy group meetings that was co-led by Ambuj and Melinda and Lourdes that I brought up this idea and had incredible support from Canadian patients and caregivers. At C3, we believe research is care, that every patient deserves an opportunity to participate in a research study. And maybe that research study itself is not considered standard of care, but the opportunity to participate in research should be considered standard of care and should be offered to every patient. We believe that research brings hope, and we want to use that to translate into better outcomes by getting patients better access to personalized therapy, expert opinions, targeted agents, immunotherapy, and of course a pipeline of preclinical research. And there's a picture here of Dr. Leonard Anka, who's our C3 project manager. He's been there from the very beginning in the grant writing process, and a lot of the work that you see has been led by him. So the first aim is the one that I lead. It's the HOPE aim. And really what we heard from Canadian patients and caregivers is they felt very isolated. Remember that Canada is geographically bigger than the US, but we have about 1/10 of the population. And so some patients really felt that their oncologist maybe had not treated a patient with cholangiocarcinoma for years. And some even said, I think I was the only person in my city with cholangiocarcinoma. That's how they felt. So we were really trying to bridge this by creating a community, much like the community for the Cholangiocarcinoma Foundation, but unique to Canadians and for Canadian problems and solutions. And Christine Lafontaine is one of the key people, and she's here as well, who leads this as our research navigator. So she is the primary contact for every patient who connects with our organization, C3. And what we wanted to do is get patients access to molecular testing. We wanted Canadian patients to be able to participate in the patient registry. I'll talk a bit about that. We wanted patients to have access to a national multidisciplinary rounds with experts, Canadian experts, so that they didn't feel that their care was being looked after by someone who didn't understand the disease, that they had access to researchers who look after many of these patients. And also understand all of this as part of a HOPE study. In addition, clinical trial matching, and thanks to the Cholangiocarcinoma Foundation, we were able to provide Canadians care kits, much like the Americans can receive through the foundation. So we had to start by building a network of physicians across Canada. We're still working on this, but we really wanted to try and connect with every single oncologist who treats patients with cholangiocarcinoma, because that would allow us to connect with every patient. So far, we've connected with 117 medical experts across all the provinces, not the territories, although much of their care would be done south anyway. And so this has been a great group to be able to spread our information to, and also get input on what their needs are as the physicians who look after these patients. We had to reach out to patients, so we created a website, cholangio.ca. And it's really more than a website. It's linked to the foundation's website as well. And I'm super grateful for the foundation who were able to translate some of their materials into French so that our French patients can also have access to that important information. It has a list of the medical experts, resources for patients, and an invitation to join the C3 where they can access things like molecular testing, the registry rounds, etc. And then we have our presence on social media. So I showed you this slide and how exciting things are, and particularly in cholangiocarcinoma. So this is just a list of some of the mutations where there are drugs currently available. I won't go through them. There's other sessions where you've learned a lot about that, and many of you patients are more expert than I am at all of this. But what we see that in Canada, we have less than 1,000 patients a year diagnosed. So if you have a mutation that's present in 1%, that's about 10 Canadians a year, which makes it challenging to identify those patients and also challenging to have a voice. And here's the current state of implementation of these discoveries in Canada is that there is no province that has comprehensive molecular testing that's funded in Canada. And we have no insurance companies that fund it either. So this is really a huge problem, and this is why the C3 really wanted to address this problem by providing patients with molecular testing. You can get molecular testing if you pay out of pocket. It's between $2,500 to $5,000, and that's not affordable to many Canadians. Even if you do get molecular testing, it's difficult to get into any clinical trial. We have very few site-specific, so cholangio-specific clinical trials in the country. We have a few that are site-agnostic, so just mutation-specific, but even then, certainly not that many. And because we're geographically very large, if it's open in one center like downtown Toronto, that's a pretty huge travel to do. You can sometimes get compassionate access. You can pay off-label, very expensive as you can imagine, or you can do what most Canadians do and not get access at all. And then because, again, we have such a big country with geographically big with a small population, it's very challenging for us to run clinical trials without some kind of decentralized or regional or virtual option for those trials. So I'm very pleased to say we are now able to offer molecular testing for Canadian patients through a standard of care route. We have a contract with OncoHelix as well as a laboratory in Toronto, and we're just launching that. Took a while to get contracts and stuff. And we also have a research opportunity for patients who are having surgery to get whole genome sequencing that's linked into a collaboration we have with the Ontario Institute of Cancer Research and Terry Fox. Canadian patients, in theory, can access the registry, but they have to be able to procure and upload their own records. And I can tell you that this is quite challenging. In the US, there's a law that you don't have to pay for your own medical records. That doesn't exist in Canada. And each province has its own rules around access. So patients can be charged for their records, and it can be very challenging to get them. In the US, a citizen is able to procure records on a patient behalf, and that's not true in Canada. So we are working with Citizen. They've been an amazing partner to try and sort this out in Canada so that patients can have access to the registry, which would allow them to have a virtual chart so they could go to see if they can be eligible for a clinical trial or to get a second opinion. It'll also be critical for us to have real-world data in Canada and also to merge with the other international data to get real-world evidence. We have started a national multidisciplinary rounds, and one of the key features here is that patients can ask for their case to be presented. It doesn't only have to be their physician. Patients are not invited to the rounds, but we have experts in medical radiation and surgical oncology. We also have a dedicated pathologist, radiologist, and molecular oncologist to review all of the cases. And we're able to provide records back to the patient and their treating physician and what the recommendations are. We've also partnered with the Canadian Cancer Clinical Trials Network for clinical trial matching. One of the issues that we've realized is there really are not enough clinical trials in Canada. And so patients who want to participate often have to leave and pay out of pocket in the US to participate, which is obviously not ideal. We've also partnered with Auer to be able to provide some of the travel expenses for clinical trial participation. And we're also measuring hope through this process. So we want to understand what cholangiocarcinoma patients hope for, how we can provide it, and then whether our interventions are improving hope. The last two aims, I'll just quickly say we are in the process of starting a TIL therapy trial in Canada. We are manufacturing our own T cells in Canada. This will be the first academic manufacturing of T cells in Canada at the CHUM, and we hope to have that trial up and running by 2025. And then we have a pipeline of experts across the country who are working on developing particularly novel immunotherapies that we can combine with existing therapies and with T-cell therapies to improve outcomes in the future. Public advocacy is something we've also been able to do. We have our Canadian Advocates meeting monthly, and we've done letter writing to provincial and national funding agencies, advocacy on Parliament Hill, published op-eds around some of these issues, and there's a picture in the corner of Julie, who's one of our Francophone patient partners who's really been active in the Francophone media and has been profiled in one of our national magazines, Maclean's. And again, partnering with patients and other organizations in Canada to just improve awareness. There's a picture of one of our patient advocates who's here, Cynthia, who is profiled on CBC, which is our national television network, raising awareness on World Cholangiocarcinoma Day. So this is our contact information, and really want to say thank you to our funding bodies, Canadian Cancer Society and CIHR, to the foundation in particular for being so supportive in helping us get started. And I think it's important to recognize that they're not just assisting patients and researchers and physicians, but in our case, an entire nation trying to improve care. And lastly, but not least, to my husband, who is my source of inspiration every day, and to all the patients here, your resilience, your dedication, your grace, it really does inspire me to try harder and do better every day. So thank you.
Do we have time for questions? We'll take a couple questions if anyone has questions for this panel. If not, you guys can reach them. Like Ryan said, there is a table right out the door to the right for the PECGS. The GWAS room is down the hall, and C3 also has a table in the hall. Yeah. One question? Okay.
Hi, I just have I'm not quite clear about the GWAS and what happens with the, like once your blood is taken, then what happens? And does the patient or the donor get any feedback in some way, or I don't really understand it.
Yeah, that's a great question. Thanks for asking. So currently what we're doing is we're taking blood, isolating DNA, and then de-identifying it for it to be sequenced and analyzed. So we're looking really at the population level right now as opposed to an individual level. The data that we're getting out of this study isn't directly applicable to you as an individual patient, and that is certainly an area that needs to be further explored. That's why I said it's really a paying it forward thing. The more we learn about genomics at a population level, the more it will be able to be applied to individuals in the future.
Anyone else?
Can I just make one quick comment? I think one of the things that all three of our research projects show you is something that's really a paradigm shift probably over the past 10, 20 years, whereas you wouldn't have seen research like this get funded by any agency years past, where people are truly trying to partner with patients and patient advocacy groups. It was very much the doctors were here and the scientists were here, and we would get samples and study things, but this patient engagement and this looking at different ways to break down barriers is a relatively new concept, I think, that's obviously long overdue. But you're seeing three projects here that are sort of overlapping but attacking different areas that I think represent the future of us all trying to figure out how to get the right treatment to the right patient at the right time, and if the patients live far away or the patients, there's barriers, breaking down those barriers. So it's a great thing to be a part of and to see, and we couldn't do it without all of you, without the foundation. So thank you.
Exactly that patient-powered research we talked about. Yeah. Thanks, panel. We appreciate it so much.